So, good morning, everybody, and good week also. So, most of the lecture today, most of the class today will be devoted to the second part where this is a bit, is the lighting good? I think we don't want, no, yeah. It's good like this? Not too, okay, not too dark. So today we'll end the part about the main olfactory system, and then for, I hope, most of the time, we'll talk about the other system that I want to talk about, the glomerulonasal system. So if you remember, I hope you do, let's start immediately with this slide. We've ended with this slide, right? And what was the main point of this slide here? Main point of this slide was that microcells, what?
Different mitral cells have different...
Right, that individual mitral cells which are connected to the same glomerulus may have slightly different activity profiles or response profiles, but they do share they have very similar response properties to those you would expect from the receptors, right, that they connect. This was the main point of the slide, and then we discussed, or we mentioned, the fact that different mitral cells may, if we look in a more detailed manner, we'll see differences in responses to a large set of different stimuli. Okay, this was the point over there. And the next thing, which I don't think I really gave time to look at this slide, was to try to explain some of these differences, and we'll explain them in a very superficial way. So what you see here is again the olfactory bulb, right? This is a coronal section. Remember, we're cutting the bulb. This is the midline right here. And what are these two things that are colored? Glomeruli, which were in this specific experiment labeled. And this is a blow up of one part. This is it has the same structure around the entire circumference of the bulb on each of the two parts. And if we look at one such section, we'll see that there are different layers. It's a little bit, looks a little bit like the cortical layering, but it's not the cortical layering, it's a different layering. And we have several layers. We have the glomerulus, the glomerular layer, and these things right here in the center are the glomeruli. What happens in the glomeruli? What do we have? Who talks to who? Right, we have the axons of the sensory neurons, and we have the dendrites of the mitral cells, and they're here in the glomeruli. And the reason it seems so white is because there are not that many cells within the glomeruli. What defines them in these images are actually the cells around those glomeruli. And one of the papers that will be described talks about some of these cells, about a population of these cells and their roles. This here is a very important layer. It's called the mitral cell layer. So this is where the cell bodies of the mitral cells are. And there's another layer called the granule cell layer. You don't have to remember all the layers. I just want to point out the important ones. The granule cell layer is the one, it's actually the center, or the central part of the bulbs, which contains the most abundant cell types, which are the granule cells, which may not seem that important because they don't project outside, but they're very prominent interneurons that shape the responses of the mitral cells. Okay? And here you see a simplified schematic description of this circuit. So again, you have this is a flipped version of this image, and very simplified, as I said. So we have the sensory neurons, we have the mitral cells. These would be the apical dendrites, cell bodies in this mitral cell layer, and the axons, which reach. Where do they reach? You don't know yet. They reach other regions in the brain, like the olfactory cortex. And there are two interneuron types, which I mentioned: the granule cells right here, and the periglomerular cells. Okay, so this is the basic diagram of the wiring in the olfactory bulb. And if you look at more detail, and this is a more detailed representation, you see that there are more cell types than what I've just described. And of course, there are always subdivisions, right? Mitral cells are actually mitral and tufted cells, and depending on where they are and what connectivity they have, you have different subpopulations, and the same is true, even more so, for the different interneurons types. We won't talk about them. Also, we don't know what all of them do. I mean, there are many things that could be done, and we don't know the roles of each of these interneuron classes. Some things that we do know that they do is, for example, is they help with the process of concentration invariance, which we mentioned when we talked about very early on about what kind of thing we would want a robot to be able to do, right? Remember to identify orders regardless of the concentration. Another thing is state-dependent processing, which we also mentioned, which is how our internal state or the animal's internal state modulates information transfer already at this early central processing stage. And this is largely dependent on inputs that come from other parts of the brain. Okay, so we have also incoming fibers from various regions of the brain. And plasticity and learning will have one, there is one example of learning that I will give to you, a nice one in the other system, in the other olfactory system. But the general layout holds both for this, the main olfactory system, and for the vomeronasal system, which I'll discuss later. Okay. One thing that I'd like to mention, especially in light of one of the papers that you'll discuss, is the fact that stimulus axis on olfaction is coupled, of course, to the action of breathing, right? It's something that happens all the time, and therefore there is some inherent timing to the act of stimulus uptake in breathing. And what you see here, I don't want to get into the details. I just want to show you the general coupling between inhalation and exhalation and responses in the main olfactory system. But this is a graph that shows you the inhalation of a rat. And you can see that there are very distinct phases, right? This is a very slow sniffing phase, and this is a very fast one. And what do you think is happening during this very fast one? What kind of behavior does this correspond to?
He's running.
What? Okay, but he's not running in this case. It could be that he's running, but right, it's active sensation, it's active sniffing. I think it could be probably running as well. I don't know how they look like when they do an effort test, the rats. But this is an active exploration of some target. In rats and mice do that, and they're actually different. If you look at the distribution of the rates of the sniffing, they vary very much. I think we discussed this issue, right, about the frequency of breathing. I think we raised it, right? Yeah, and so I don't know if it was in front of everybody or not, but it can be pretty fast. It can get to the range of 10 hertz, which is very fast, right? Breathing 10 times per second. Not in humans, but in smaller rodents. And what you see here are the responses of sensory neurons. So this is a sensory neuron here, just some kind of a profile of, I think it may be an imaging study actually, not a direct electrophysiological measure. What you see here is the respiration. So there's some monitor of the inhalation and exhalation, and you can see that when an odor is given, this is this horizontal bar right here, the responses in the sensory neurons are coupled to the breathing, okay, as you might have expected. And what you see here is one stage further, the stage of the mitral cells in the olfactory bulb, and again you see the same thing. So this is respiration. It shows the same data or the same variable, the same parameter, which is breathing, but it's a different measure. So this and this looks different. And here you see the mitral cell responses, and you see the action potentials which occur. It's very clear here that there's some rhythm, right, that's locked to breathing. So it's not a continuous phasic response. It's coupled to the breathing. And indeed, when you look at responses in the olfactory system, you always, and the brain probably always has to somehow compare them or consider them in light of the ongoing breathing cycle. And here's one example of how this really matters for representing information about stimuli. What you see here is a response of one neuron measured to 5 different odorants. One of them is a blank odorant, and these are 4 different odors whose names are not written here right now. And each of these, each line here responds to one trial repetition. Is this a familiar representation to you all? The raster displays, okay. And what we see here are the PSTHs. So these are the average firing rates. And time 0 is the time of the start of inhalation. That's an important aspect to understand these figures. Now, this shows you the actual firing rate modulation as a function of time since sniffing onset. Okay, and this shows you the mean change in firing rate. So if you take the whole first cycle of breathing and you just sum the area, if you will, under these curves, the number of spikes that occurred following odor presentation. So the point here is that if you look just at the firing rate, you may have a hard time distinguishing between, for example, the blue and the green odor right here, A and B, right? They have very similar firing rates. On the other hand, if we look at the shape of, or if we look at the modulation of the firing rate as a function of time, we will see a difference. And presumably, and we know actually, that the downstream processing stages have the capacity to take this kind of information into account and use it to distinguish between different odors. Okay? So the point here is it's not just the firing rate over a long window that matter, but also the precise change in firing rate as a function of time locked to what? What is the important thing to register the responses to? To the breathing, right, to the sniffing.
What about the difference modulation in time?
This is a good question, and it could depend on many things. It could depend on it could depend on the interactions within the bulb itself. Okay, so you may have some receptors or some odorants which also activate other neurons, and those interactions. And another factor is also the chemistry of the odorants may also make a difference. So the way that they activate the sensory neurons may differ for different odors. Okay, the physical properties. And this is a generalization of what we just saw here. Here we see different neurons and the same or some of the different odors, and we see that a given neuron may have different responses, different temporal responses to different odors, which is what we saw here. And at the same time, a different odor may evoke different temporal patterns in different individual mitral cells. Oh, yeah?
Does the bulb have any motor input about the breathing? Like if any?
It's a good question, and it's not entirely known if there is direct motor input. And one of the questions is what exactly causes this ongoing modulation. So the answer is not known exactly about the motor input. But definitely this information exists in the brain. Maybe it's not necessary because the incoming information, there are actually sensory neurons which are sensitive to the airflow, not just the odors. You see that often that even a blank induces a response. And so this may be sufficient as information for these. But maybe not the olfactory bulb, but downstream regions must have this kind of information. Okay, so this is just saying in a more concise way what I just told you right now about the different responses in different neurons and for different odors. Okay, yeah.
We know from other senses that there are like ways that we can sense and others that we cannot, like in vision or in auditory.
That what we can sense? Wavelength,
Like
you mean, or
certain
frequencies?
Mm-hmm. So is there something to that in olfaction?
Yeah,
like there's a scale or something.
Is there, so in vision and in audition, it's simple. There's one parameter, right? There's the frequency of light or the frequency of the pressure, of the air pressure. In olfaction, there's no single parameter. That's something that we discussed. The olfactory world is I don't know how many dimensions there are, but so we cannot say, it's from here on to here on we cannot smell things clearly. I don't think we can look at an olfactory space and predict based on the properties of the molecules whether it's going to be detectable or not. But of course there are things we cannot detect, and that depends, first of all, we mentioned this on their volatility. So that is a chemical feature actually that we can predict whether it's volatile or not. But the other thing is whether we have receptors that can actually detect them. And definitely there are molecules that we cannot detect. Yeah. just solved the puzzle when there was one question: what's common to CO2 and natural gas? And the answer was odorless. They're both odorless. So there are indeed things that they could be detected chemically. They're molecules, but we don't have the receptors to detect them. Okay. So now we go on to the next stage. And the next stage, there are actually multiple next stages. Actually, I'm missing a slide that I usually show, which unfortunately I don't have here. But that slide shows that if you look at the axons of the microcells, they project to various regions in the brain. Okay, there's the olfactory tubercle. Maybe I'll add that slide there to the presentation that I'll give you. But there are several regions in the amygdala, in the cortex, and the one of the key regions, the one that's most studied and it's also a prominent region in terms of its size, is the piriform cortex, and it's usually thought of as the primary olfactory cortex. Okay, and you can see it. Actually, you have this very simple diagram here. That's not what I'm talking about, but you see here that telling you where the projections of the mitral cells reach. I'm not even going to tell you what each of these mean, but the point is that there is a widespread divergence of outputs from the bulb. And actually, that's an important point that I don't think I said explicitly. The mitral cells, they're the ones who receive the information directly, and they are the ones who send all the information to these next regions. And so now I'd like to take a look at this region, or the responses in this region, which is called the piriform cortex, and look at the presumably responses of pyramidal cells in this region. I should also say that this is not a neocortex. It's not a classical 6-layered cortex, but it's an evolutionary older cortex which has 3 different layers, so it's simpler in its organization. And so let's look at responses to odorants in the olfactory cortex. We looked at sensory neurons a little bit. We looked at mitral cells a little bit. And what you see here is an experiment that used calcium imaging to look at broad regions of the cortex. So again, you see the region that's being visualized, imaged, and these are the imaging planes which are superposed together to show you the general picture. And each of these little things, of course, is a cell, and when it's red, it means that there was a strong enough calcium signal indicating that it was active. And what you see here are responses to 4 different stimuli which are monomolecular odorants. So pinene, cadaverine, octanol, and butyric acid. And you can see that the responses are very widespread. Even to a given single molecule, you have very widespread responses, and we don't see any organization, any obvious pattern like we did see, for example, in the case of the olfactory bulb. So this is one thing. We see a lot of a very broad pattern of activity. And the question is whether individual neurons, if we look at them, they also respond to multiple odorants. We cannot tell from this image because we don't know, or it's hard to see the correspondence between different neurons, even though it's the same imaging plane in these 4 stimuli. And if you do this, if you superpose the responses to different odors, you see that indeed there is overlap. So there are many cells, or there's a considerable fraction of cells that respond to more than one stimuli. You can see it here in these images. The neurons that respond to octanol are shown in red, and in each different panel you have also the responses in green to one of 5 other different stimuli. Here's a blow-up actually it's not a blow-up, these are different odorants, and you can see that there are some cases, not a lot, but there are some cases where you have these yellow cells indicating that they responded to both of the stimuli. So at the level of the piriform cortex, the responses are not highly selective. A given neuron may respond to more than one monomolecular odorant. Okay. And now, we can ask ourselves what kind of connectivity from the bulb would account for these kinds of responses. Okay, it's a very, there's more than one answer, of course, but connectivity, if we look at the axons of mitral cells, okay, let's consider one individual mitral cell. Suppose we label its axon. What will it look like when it reaches the olfactory cortex? Remember, we asked the same question in the case of the sensory neurons, right? And we saw that they converge to glomeruli. So does anybody want to take a guess? What would we see if we label one mitral cell? Yes. And this is why I don't like to give you the presentations ahead of time, because then you would know, but you don't have.
Can you give us some numbers, maybe, like the number of mitral cells?
I can give you some numbers, yes, but numbers of what? What are you asking about?
I don't know. Like, would it be 1 to 1, or maybe a single mitral cells and maybe too many neurons? I don't know.
Okay, so I'll give you some numbers. Yeah. So first of all, let's look at what happens when you label one mitral cell. Okay, and this is an image of one mitral cell labeled, and you see here 3 different views. Okay? It's the same data, but 3 different views. So this is the most intuitive one, I think, to look at. The lateral view. This is the olfactory bulb. This is one mitral cell, and I think it's really striking how profuse the projection from one neuron is, how it reaches so many different, such a broad region of the olfactory cortex. And you can imagine that we have different cells like this. They will, of course, overlap each other, right? Because they're not, but neither of them is circumcised to a small region. Now, the numbers we talk about a given mitral cell, usually it's actually written here as about tens of thousands of pyramidal cells may receive information from one mitral cell. 1 second. And a given pyramidal neuron in the cortex may receive information of about 100 or so different mitral cells. So it's a large number, and it can be a substrate for being sensitive to what? What can a given pyramidal be sensitive to? Yes, to combinations of odorants, and that gives them the ability to. We started out talking about the fact that odor objects are complex mixtures of molecules often, and this connectivity gives the pyramidal cells the ability to be sensitive to different combinations. And we can imagine that even combinations that have never been encountered before, they will have some pyramidal cells which are sensitive to these specific combinations. And you had a question? Not anymore. Okay. So this kind of data gives us our current view of the piriform cortex as some integrator region that can be sensitive to combinations of odorants, and also explains, or this is also corresponds to our thinking that a given odor object is represented by a combination of pyramidal cells, each of which itself can be sensitive to a given combination of single molecules. So this ends the part about the main olfactory system. I think we're actually on pretty good time. So let me just summarize this. It's not, yeah.
I'm not sure I understand the idea of the combinatoric. So we're saying because the olfactory cortex can have input from different mitral cells.
Yes.
But they're also sensitive to different odorants. So like integration in both levels.
It's integration in multiple levels, because a given mitral cell can respond to multiple stimuli, and a given pyramidal cell can. Yes, it is. We don't exactly know how really odors are represented, but the evidence that we have suggests that the representation is some distributed pattern of activity of a specific subset of pyramidal cells. This is our current understanding. Ken? Yeah.
The concentration of the different concentration of another, like there's a lot of molecules, the variance in this concentration gives similar patterns in the firing, right?
Where? Sorry.
In the sensor at the beginning, like you get the same matter but in different concentrations.
No,
of the same molecules.
It will be different for a given molecule at different concentrations. You will see a different response. There's usually and it's somewhat normalized at the level of the olfactory bulb. And it gets more complicated in the cortex because you have some cells that are more responsive to the odor, to the concentration, and some which are less responsive to the concentration. But these are hard questions. Not hard, but these are the questions that I don't know. This is where we're still trying to understand how the system works in terms of representing information. Okay. Yeah.
Do we know which type of computation or memory recalling is done in the bulb itself versus the cortex?
So some of the computations that we think of are, I mentioned this several times, is the concentration invariance, Another thing is plasticity related to probably to suppression of specific mitral cells. I will mention this in the next slide. Lateral inhibition is something that may happen to some extent in the olfactory bulb, so sharpening of certain representations. We don't know a lot about what happens in the olfactory cortex. So these are the things that are known that I can come up right now that are known to happen there. So not a whole lot is known about the actual computations. Okay, so let me summarize before we go to the next part. One is olfaction is important. I hope I convinced you this initially. And that individual sensory neurons express one type of receptor. This is all going back to what we said. The axons reach the olfactory bulb and they converge in glomeruli, right? All sensory neurons expressing the same, or not all, but sensory neurons expressing the same receptors target two glomeruli, and there they synapse with mitral cells. Okay, one mitral cell targets one glomerulus, and we talked about the fact that mitral cells, we have what are called sister mitral cells, which target the same glomeruli. That's what we started this class with. Remember the number of different mitral cells, order of magnitude, that target a given glomerulus? What? Yeah, 50, 40, something like this is indeed the number. This is where we started today. So similar responses to sister mitral cells, but not exactly identical. Information is relayed to the cortex in very diverging connections, the yellow labeling that we saw now, and the fact that the responses are widespread and that this allows the cortex to encode arbitrary combinations of molecules. And of course, there may be an order that is there, but our ability to view it doesn't uncover it. And the next slide, which is not a summary because this is actually the jumping stage to the next part, is the fact that we actually have multiple divisions in the main olfactory system and generally in chemosensation, and there's not just one homogeneous type of sensory neurons at the first stage. So this is, now we move on to the second part. I don't want to take a break now. I want to take a break after 45 minutes. Chemosensations, part 2. מה זה? מה? זה לא מה? לא. לא. לא שמתי את זה. I will put all the presentations though at the end of the class today. Okay, I mean sometime at noon when I reach the medical school. Okay, so now we talked about did you get the other presentation? No.
Yeah,
Oh, you did get it because Mati said you won't put it up. Okay. I said that. Okay. And you like having it, right?
Yeah, because then you can write them.
I know the pros, but I also know the cons, which are the fact that. So even in the first part, you had everything on before. What? Yeah. Okay, you didn't look. Okay. It doesn't matter now. Okay, so no, this one is not there. Okay, so the starting point is that chemical communication is very important, right, in the animal kingdom. We're not talking just about humans. And often we use the term pheromone a lot. Does somebody want to say what a pheromone is?
Like a molecule that acts as a communicator between individuals.
Okay.
Will affect behavior.
And affect behavior. So yeah, I don't want to get all the different possible interpretations of the word pheromone, but the classic one, the one that was conceived initially, is that it's some compound, some molecule, a single molecule that acts within a species. Okay, that's part of the definitions, and one individual releases it, and another individual detects it, and it elicits some kind of a response. And the response can be physiological or behavioral. And the classic example is and actually all of the very clear examples are examples in insects, which are simpler organisms. And there, it's more likely to find a molecule that elicits some kind of a very stereotypic behavior. And this is the example of the molecule called bombicol, which was identified and with a heroic set of experiments isolated, and its structure was found. And this is a molecule that the female silk moth releases, and the male detects it. The male has very elaborate antennas, which the female doesn't have, and that gives the male the sensitivity to this pheromone and allows him to find the female also from a very distant location and fly towards her and have baby moths eventually. And yeah, so this is the classic example of a pheromone. But it's important to remember that pheromones are not the only type of semiochemical. So pheromones is not the only type of communication that can play a role between different animals in nature. And so let's look at the general classification. So we have semiochemicals, that's the broadest class. And we have those that act within a species. One of them, of course, is a pheromone, which we just saw. And we have those that act among species. And here, within a species, we speak of pheromones as one class. And the thing about pheromones is that they're not individualized. So there's nothing unique about one female's pheromones versus another female's pheromones. They're all identical. They may have different abundance, but it's the same molecule, and it has a clear, unambiguous meaning to the male. And within, again, we also classify pheromones into two types. One of them is releaser pheromones. And again, these definitions are most appropriate in insects. Okay? Releaser means that it releases some kind of behavior. So the pheromone that we just saw is an example of a releaser pheromone, because the behavior that was released was what? Initially, at least? The flying behavior towards the female, and eventually the mating behavior. But I think the pheromone itself induces the flying actually towards the female. And this is this example. The other class is the primer pheromone, primer effect, and that means that it primes, it changes some physiological process. So this is something that, or hormonal process, something that may not be visible looking at an organism, but it changes something about the physiology and will have an effect later. And this is an example of one process that I'll actually mention, so I'm not going to this is called the Bruce effect, this example right here. I will mention it in a few slides. Then we also have within a species what are called signature mixes, and they are different. They're unique identity profiles that individuals may have, and they're used to distinguish one individual from another, and they're very important in many animals. For example, this is an example of a lamb, and the ewe, when the lamb gives birth, she recognizes the odors of her offspring, and that helps her to identify him or her and distinguish it from a stranger. And this is one example that there is something unique about the smells of a given individual, and it's not a pheromone. Okay, it's important to remember. We'll give some examples of these of the molecules that can be such signature mixes when we talk a few slides further down.
The difference between this is not individual, this individual, or the
Yes. So this has some general meaning, and this exactly. This is the difference. Okay. Now let's talk. Yeah.
the pheromones only elicit the ones that are connected with mating, or
Pheromones?
yeah,
No, not necessarily. Not necessarily. They can also elicit responses that have to do, for example, with changing developmental stages. If we talk about insects, people often talk about. Again, it depends how broad you are with the definition of pheromones, but there are stimuli that can elicit aggression in other animals, for example. So it's not just about mating, but mating is it's an important part. And usually, when we talk about pheromones, we think about mating, but it's not only about mating. On the other hand, the repertoire is mating, fighting, offspring, taking care of offspring. So it's not like there's an infinite range of different behaviors. It's pretty limited. Okay, now The other broad definition is those chemicals that act among species, so between two individuals which are not the same species. One example, and there are many, this is a very common type of a molecule, are classified as allomones, and they have a benefit to the signaler. So for example, if I'm a predator and I have some bait, then you would consider that molecule that I elicit as an allamone. And in this case, this is a very nice example of a nice, not so nice to the prey here, but you have a spider. It's a South American spider. It's called the bola spider, and the female doesn't build a net, but she does have on the silk. She actually has a little ball, sticky ball, and in addition to the sticky ball that hangs on a wire, she also emits a molecule that mimics the pheromone of the female moth, and that causes the male to be attracted to the spider, and the spider catches him with this little sticky ball and eats him. So this is an example of an allomone, of a bait. Chiromone is the case where something is of benefit to the receiver. So for example, a tick. This is a deer tick that can identify the location of deers, and then based on their orders can latch onto them and suck their blood. So this is, in a given molecule can be either an allomone or a chiromone. It can play different roles in different contexts. And finally, there's actually some argument whether there are really cinnamones, which are supposed to be molecules that everybody benefits from them. Usually, always somebody benefits more than the other in almost every kind of interaction. But the example that's given is between mites and between ants. But it's not actually. There's some dispute whether it really is a synergism or not. So the major classes are all of those, everything except the cinnamones. Okay. Yeah.
Communication between animals and plants also are included in these categories?
I don't think. They could. Yeah, but yeah, I guess they could. You could put them here. Yeah, plants and animals. Yeah. You could. Yeah, and also plants and plants, I guess. Okay. So we gave some general examples or some general classification, and now I'd like to show you just some example of how important chemical communication is how rich are the different methods of advertising chemical information. So this is an example of a mara male, which is a South American rodent, and he's peeing on the female to advertise probably his strength and his health status. So this is one kind of way to advertise your strength. This is an example of deer which are depositing their smells, their scents from glands that are located on the facial region, and this is actually the kind of information that can aid the tick that we saw in the previous slide. So the point is that animals also rub their secretions. Cats do it, deers do it, many animals can do this, and there are also specialized glands in order to produce these chemicals. And this is a very extreme example of it's called a wing sac bat. I think it's also South American. You have a lot of these South American examples here. And what the male has is a little kind of sac on the wing, and it's the sac serves to collect all kinds of different secretions like saliva and feces and urine, and it's all mixed there, and it ferments probably. There's some stew that the male makes, and then he uses his wings to blow the scent over the female. So it's another example of advertising in an active way. And of course, even if we think of this as something that's maybe a little bit disagreeable, you can think that there's a lot of information probably contained in these secretions that the male advertises to the female. And finally, here's an example from humans. We all know the use of perfume, and that it's something that definitely has a role in interactions between different humans. Both males, of course, and females use perfume. And also the point is that it's a different form of we don't spray perfume on other people like the example here or here, but it diffuses through the air in a passive way. Okay. So this was about the actual signals, and now we can start talking about the different about the mechanisms of detection. And what this slide shows you, what you see here is the olfactory bulb, and each color represents a different type of receptor class. So we won't go into the details, but so far we talked about the different, or I mentioned the different olfactory receptors. I mentioned that there are about 1,000 different ones in mice, right? Active sensory neurons. And what this shows us here, that there are actually different regions of the bulb that receive different types or information from different types of sensory neurons. Again, we won't go into the different classifications, but they may differ in what kind of molecules they secrete, even in the signal transduction pathways within the olfactory sensory neurons. And what this shows, again, the details are not important, but this is the genetic relationship between the different receptors, which are almost all of them, not all of them, but almost all of them are G protein coupled receptors, and they have different sequences, of course, and some of them are more similar to each other, and some of them are less similar to each other, and this is just a tree that shows the relationships between them. The point is just that there is a correspondence between the different receptor types and some regions on the bulb that receive this information. And some of these regions on the bulb, like this one here, which we'll talk about a lot, do not receive the information from the main olfactory epithelium, but rather from other regions. Yeah.
But this map is based on spatial similarities?
On sequence similarities. Not on structural similarity, because we don't know the actual 3D structure for most of them. Okay, so this is what happens on the olfactory bulb. In the next 4 slides, and then we'll take a break, I want to show you that there are different epithelial locations, or different locations of the sensory neurons that may project to these regions. So this is the main olfactory epithelium. This is the main olfactory bulb, and within here you have different classes of sensory neurons, which are shown in different colors in the previous slide. Never mind the different subdivisions. A very important system, this is the one that we'll talk about after the break, is it's abbreviated here as the AOS, the accessory olfactory system. I usually refer to it as the glomerulonasal system, or VNS. And you see that the sensory neurons, first of all, are not in the main olfactory epithelium, right? They're not here. They're not where the yellow part here. They're in a different region, which we'll talk about. And they don't project to the whole bulb. That they project to a region in the posterior end of the olfactory bulb. I'm going over this quickly because we'll discuss it in more detail. And I'm going over this quickly because we won't discuss it at all, and these are more minor systems. But I'd just like to mention that there's also a subset of sensory neurons which are not really where the main olfactory epithelium is, and this is called the septal organ, this little patch here, and it projects to a certain location in the olfactory bulb. And you have another set of neurons. This is called the Grunewald ganglion. It's very close to the nostril, and it's a small subset of cells that projects also to you see this region. It's close to where the cells of the accessory olfactory system project to, but it's a distinct set of glomeruli that receive this information.
Do the instruction systems exist in humans as well, or just
most of them, no. We don't have the Grunewald ganglion. There is no active accessory olfactory system. At least, definitely, it's not similar. There may be something like the septal organ. I'm not sure if there is or not, or some set of cells. And the different divisions of the different divisions, I think they do exist to some extent in humans. Not the whole range that you see in mice, I think, but there are some of these divisions. Finally, I'd just like to mention this system, which it's the trigeminal system, which is not really an olfactory system, but it's a sensory system that senses pain and chemo and other aspects of chemicals. We talked about this when I was asked about innate avoidance of some odorants. Some of the things that we may perceive as inherently very unpleasant, like acid smells or very strong smells, they may actually act by activating the trigeminal system, which is more often associated with pain, rather than the sensory neurons of the olfactory system. So we also have sensory endings in our nasal cavity that are sensitive to.
But they're just sensitive to the odors, but you don't perceive it as a smell, right?
We don't perceive it as a smell, it's true, yeah. But they can elicit a response like tearing or some unpleasant response. Yeah, but it doesn't help you to distinguish one smell from another. And it may also be that you may be able, if this system is gone, you may be able to distinguish the odorants, but they may not elicit the same kind of aversive response. And at least in mice, we know that such divisions can be made. There are some sensory neurons. Never mind, actually. I don't want to get into that. Okay. They are. They're at the front of the nose, and very close to the nostrils. So let's take a break here and come back in 15 minutes. כן.
כאילו, אתה יודע, באיזשהו שלב שהביטים הם שולחים שהם שולחים אקסונים
פירפון.
כן, לפקטורים קורטקס.
כן.
אבל לא, כאילו, לא הגיוני יותר שהם ישלחו לפקטורים בלב בעקבות משלם?
רגע, אבל אתה מה הייתה להם באלפקטורים בלב?
הבנתי. הם היחידת טיפול, לא מה שמחזיק אליהם מידע?
לא, התאים הסנסוריים הם אתה יכול לראות את זה אפילו. אלה התאים הסנסוריים שמגיעים לאלפקטורים בלב, ופה יש את התאים המיטרליים, מקבלים את המידע הסנסורי ושולחים אותו הלאה. אני חושב שעשית איזה קפיצה כן, התאים המיטרליים זה התאים באולפקטורים בלב. כן. אבל דווקא שאלו אותי את השאלה ההפוכה בשיעור שעבר, שאלו אותי, לא יותר הגיוני שיגיע התאים הסנסוריים ישר לקורטקס?
לא, זה לא הגיוני. מה יש לנו את האלפקטורים בלב?
נכון. אם כבר יש אותו, אז כדאי ש...
מי ששאל את השאלה, זה חסר. תודה רבה. כי הוא איזה רכב מכוון, מי שכאילו הביא את ההדקה שלו אתמול, לא יודע, 10 בלילה.
אני בטוח שתגיד 2, משהו. אוקיי, 10.
זה המשיך עד 4. אז כאילו באיזה שלב עברתי לחדר של שותף שלי, והגענו.
זה עוד קורה? זה כמעט לא קורה הדברים האלה.
אני חושב, מישהו זרק פה מקודם מהרע שהוציאו את השטות הזאת מחוץ לחוק. כאילו שכבר מייצרים אותם בלי הפיצ'ר.
זה בכלל איזה אוטו מאוד ישן, משהו.
של עד 4 שעות.
ואף אחד לא פרץ אליו?
6 שעות של...
אף אחד לא קרא למשטרה ש...
אני חושב שאני שקלתי לקרוא למשטרה, ואז הקרתי לה... כאילו בהתחלה שמתי עצמים, ואז הספקתי לשמוע אותו בלי שאי אפשר שאני עצמים. עברתי לחדר של שותף שלי.
אפשר לשמוע עצמים?
אפשר, פשוט לא הסתדר לי. לא היה לי נוח.
אני גם אפשרתי עד 4 משהו.
מה, בנסיבות יותר טובות אבל?
כן, כן.
באמת? וואלה, אני חושבתי לאיזה סיפור פיקנטי. עד 4.
לא יודע, נזכרתי על איזה משהו. אתה נזכר... כשאתה נזכר משהו שאתה לא... לא חשב אני... האמת, זה משהו מאוד שתפוק. כאילו, לא באמת נזכרתי עליו, אבל יש את העבודה שאני אומר... טוב, לא אבינו.
שלום.
אפשר שנייה הפסקה רגע?
כן? למה? להגיד להם משהו?
לא, לבדוק את המחשב.
כן? אפשר.
אבל כן. כן, קראתי את המאמר.
יש.
הורדת את הסרט לכאן או לא?
לא הורדתי.
אני צריך לעשות לקרוא קצת רקע למאמר, כאילו קצת על המערכות שלהם.
אין לא נראים שבוכים מספיק. המאמר מאוד קליל, מאוד קליל. לא כמו תורשה, באמת מאוד קליל. כן, לגמרי. כי הכל שם מוסבר היטב, אין שם הכל אין שם בדיוק. יש שם כמה אנליזות של מודל וזה שאני לא הבנתי, ויש כמה דברים שגם עד לא הבנתי על הסוף.
כן, זה לא עניין.
ויש גם תקנה מצגת כזה, זה קליק
כאילו להבין על מה אנחנו רוצים לדבר, ואז גם על מה אנחנו רוצים לקרוא לפני.
זה רגע מתמטי או... זהו, ההבנה של מה רוצים לך בדרך כלל זה הבעיה. זה מה שהיה שעה
כשאני מבין מה רוצים ממני, אז אני לא מצליח לעשות את זה, אז אני אומר, אולי רוצים ממני משהו אחר.
זה עובד? תן לו...
זה הייתה לי גם
עובד, אבל תשובה כזאת. זה הראשון.
זה הרבה מה שאתה צורך.
זה הנגן.
כן, עדיין מוריד את הממוצע.
אז אני אמר לך, לא צריך כלום. בסדר, שיהיה לך. אם אני אסביר שיהיה לך.
אני קיבלתי את זה, נראה לי שהיה לי פנים. הבוחן הראשון קיבלתי את 50. כן? מה אני אמר?
91 אופקים?
51 אופנים.
ראית את זה הוריד את הבוחן הכי גבוה.
יכול להיות שהיא באמת עשתה את זה, כי אני התפלאתי על הציון הזה. ממש נחמד, לא ציפיתי.
עדיין מוריד את הממוצע?
עדיין מוריד את הממוצע.
כמה אתה סוגר ממוצע? 200. זה יופי. אני הממוצע של סרטות היה 91. זה שנה רפואית.
ממש טוב. אני חושב שאני ארזים מקשיבים האלה, כי הבנה של הרבה
זה אין משהו אפילו. לא יודע בדיוק מה.
כן. אם אני חושב שזה מבחינה מסוים, אבל אני אהיה בחו"ל. לא, תשמע, אני מסכים. חוץ מקורסים פה. זה משהו אחר גם, הדרך למידה שלך גם במוח נוירוני וגם פה, כל כך שונה.
אני לא יודע איך אני הולך להתכונן למבחן של זה. והמתמטיקה של...
IMN?
לא, של רשתות נוירונים, גם אחרת. זה לא הוכחות של העבוד.
אני יודע, אני חושב שאני יודע, דינמיקה. זהו, אז דינמיקה באמת, ידעתי את החוק. הוא לא חומר מסובך, דינמיקה פחות. הוא היה לוקח לי הרבה זמן לעשות.
אנחנו רפואיים.
כן? אז אנחנו לא נפורי הסובלימציה, אבל לא... וגם הקונבולוציה זה לא... איך אנשים בלי רקע של מתמטיקה...
תקשיב, אני מוכר יסודיים.
לא, כן, באמת שכולם מסתדרים. גם אנשים שאין להם רקע במתמטיקה, שהם בפסיכולוגיה והם טובים. ברור שבקצות אחרות אין לו רקע בדינמיקה והייתה כאילו כותרת פרטים שם בפייסבוק מעניין?
כן, ממש מעניין. כאילו זה, בשביל זה באתי.
ממש אולי אני אקח את השנה הבאה. אני חוזר.
כשאני לא חוזר אני נהנה ממנה.
ואז אתה צריך להגיד לי איך היה סמסטר א' ברשתות ב'. אמרתי שהוא יותר קל. תקווה, ממה שאני שומע ממך הוא... קיצור, אני אקבל את כל הפידבק ממך. פיזיולוגיה ב'.
אני אביא אותך ממש של כל השטות.
כן.
חיים, אתה לא חייב לקחת את הכל?
אני תלמיד של מדעי המחשב ואני בהתמחות בחישוביות קצבית.
חטיבה.
חטיבה, בדיוק. אני טס למרילנד סמסטר הבא.
כאן, נכון.
בדיוק.
קשיף לכל
מה זה אחד? לכמה זמן?
זה חילופי סטודנטים, סמסטר.
סמסטר.
כאילו, אני עושה את זה מלא שנה הבאה, ואני צריך להשלים את זה בשנתיים. זה מה שאני אעשה.
ובמרילנד תיקח קורסים או שתהיה באיזה מעמדה?
במרילנד מדעי המחשב קורסים, כי אני בתואר ראשון כזה. ורציתי לקחת שם קורסים של חשיבות עצבית, אבל לא ראיתי שום דבר מתאים. אז דיברתי עם אלי גם, הוא אמר לי, שלח לי מלא אנשים ורפרנסים, לדבר איתם וזה, אבל שום דבר לא היה שם, כבר למדתי את זה, ועשיתי את זה, וזה לא בא...
טוב, יהיה לך מספיק על הראש גם ככה, בטח לא?
האמת, אני לא יודע, יש לי פידבק מהאנשים שם שאמרו לי שהקורסים שם הם פרקטיים, ממש פרקטיים. מישהו שהיה שם בישראל אמר לי שהוא היה יכול לעשות איזה 5 קורסים בכיף. מישהו המליץ לי, זה שהאדוויזר שם אמר לי, אף אחד לא יכול לקחת יותר מ3, ואנשים שלוקחים 3 זה אנשים נדירים שמספיקים, כאילו...
רגע, והוא אמר לך ש5 זה לא בעיה.
5 לא הייתה בעיה. זה מעניין, כאילו... אם ככה.
טוב.
בכל זאת שגם אמר לי את זה בקטע של, יש לנו המון עומס של תלמידים, וזה באמת נכון. כאילו הוא רוצה להגביל אותנו בכמות קורסים.
זה הכל בראש הרי.
זה גם משהו.
מה שאין לך זמן להיות חולה, אתה לא חולה.
סמסטר הבא זה תחילת שנה הבאה.
בדיוק, זה שנה הבאה. בואו.
גם כנראה. ראיתי כזה משהו עם זה שעליה שלי לא כאילו 100%, אולי אני לא רואה את זה. באמת לא יודעת שיש חשוב.
כאן אין שיעור בשיטות. There's no method to more, only the unit. And we have the lab only in the we have no camera. No, only the human methods, but not the first one. So we started 12. Yeah. We don't need to wake up and
זה לא משנה, עדיף שזה היה מחר ומחרתיים שעשית אותה נליגה.
אני רק כאילו כדבור זה. אני חשבתי לשאול בגלל היומן מתוד, אם יש מושג כזה. הוא יש לו כאילו,
גם ככה אני ככה טבעית ב6:30, מה זה משנה? סליחה לגמרי.
אין לך כלום. בשיטות. יש רק כאילו רק לא אצלנו. שאלת אותם
שהטקס שלך מחר, אבל זה באמת יהיה משהו
מה היא הולכת ללכת?
אתמול אני נעשתי, אתה יודע?
נחבר אותו. Find slide and we'll start. So, I mentioned several additional olfactory systems, chemosensory systems, and here is the other big one, which is called the vomeronasal system. And you see it right here. You see an image showing you a rodent's head and the organization of both of the systems. So in blue, we see the main olfactory system that we just talked about. And you see the sensory neurons, you see the projections to the bulb, and you see the projections of these mitral cells. Here they're shown going to the amygdala, but they're going to the amygdala and the piriform cortex. Actually, it's also indicated right here. This is the main olfactory epithelium. And here, the main olfactory system. And here we have the vomeronasal system, which starts at the vomeronasal epithelium. And what this image shows you is, the main point here is to show the similarities between the two systems. Okay, I'm not talking about the details yet because we will talk about these details right now. But there is a similarity of organization in both of these sensory systems in terms of the location and the general function of neurons in each stage. And one of the questions, and it's a big question, there's no clear answer, is why are two different systems needed? Why is one not enough? And the idea that people have, and even though we know it's a simplistic idea, it's the best one so far that we have, is that the main olfactory system is a generalist system, that it's supposed to be able to detect any arbitrary odorant. Of course, we know that not everything, but a wide range of odorants, and that it can learn various types of associations between odors and responses. And in contrast, the vomeronasal system is viewed as a system that's devoted for innate responses to a more limited set of cues. So we talk about the things that, the things that are associated with it are innate behaviors, social behaviors, reproductive behaviors, and aggression, and that's supposed to be the specialization of the vomeronasal system. Now you had several questions, so yeah.
Does it mean that the vomeronasal system is less plastic?
Yeah, this is one prediction from this general hypothesis. So the answer would be yes. However, in my lab, for example, we're challenging this notion of and I think that it is plastic. Well, we do know that there are various types of plasticity actually, but so this hypothesis predict that it's not very plastic, but it is plastic. So I'm presenting this hypothesis as a very simple dichotomy between the two systems. I'm not defending it very strongly. Okay. Okay.
Is it only a rodent system?
No. It's not a rodent system. I have a slide towards the end, which we probably won't reach, but showing the evolution of the sensory neurons and the sensory organ. It's not just a rodent system. Most vertebrates actually have the system. Okay, not just mammals. Humans don't have a developed glomerulonasal system, although we have some sensory receptors from the system. I hope to be able to show you one slide about this towards the end. But cats, dogs, monkeys have them. Not apes, but not human like monkeys, but apes have them. So many animals do have them, including lizards and snakes, frogs.
So we don't know its function?
What?
We don't know its function?
No, we do know that it has a function, and we do know that I'll show you some. We know a function, but we're not. We don't know for sure that it's only in innate behaviors and hardwired and inflexible. That we don't know. And I should say at the outset, the male olfactory system is also very important for social interactions. Okay, it's also important. There's overlap, but this is the simplest idea that we have about why two different systems. But as you'll see, there are also functional differences. Yeah.
So does it make the olfactory system more developed if you have this? I mean, we don't have it, and animals that we consider with better sense of smell.
Yeah.
Do you have it? So does this make it more developed or less? I don't know.
Are they more developed
They
than
more
us?
rely on information coming from smell. Does it indicate that?
I don't know if it indicates that. I don't know to say that, but there is a general idea that with the evolution, with the development of the visual system that humans and higher apes have, there comes a decline of the glomerulonasal system. I'm a little bit jumping ahead too much, but there is this general notion that we don't need it so much for social interactions anymore as other animals did. So in that sense, yes. In that sense, it means that we have less abilities that our ancestors did have. Okay, so now let's talk about the physiology of the system. There are some really some cool things about the system. This is again, it's a different rodent from the previous slide, but you see here a section, and this thing here is called the glomerulonasal organ. Okay, and you see there are two glomerulonasal organs, one on each side of the nasal septum, and here you see a coronal section. So we're making a cut like this, okay, and we're looking at the floor of the nasal cavity, and this is the midline right here, and you see that there is this bilaterally symmetric structure which contains two VNOs. So one of these tubes you're seeing right here. Okay, we see just one right here. Do you know this organ? Has anybody learned about this organ? Yeah. So, and if we look now at one side, we don't have to. I mean, they're symmetric, so we can just look at one side. It has two important compartments. One of them is the lumen, shown here in yellow, and this lumen is a space that is dead-ended on one side, and it's open on the other side. That's the side that opens into the nasal cavity. Okay, so think of it as a tube that's closed and opens to the nasal cavity. You don't see the opening right here. And there's also a blood vessel, which is shown right here in red. Okay, and the important thing is that all of this is encased in a rigid bone. And this organ actually functions like a pump. And the way we think of its function, the way what we understand about its function is that when the blood vessel expands, then it takes up more space and it constricts the lumen. And on the other hand, when the blood vessel constricts, what happens to the lumen? We have a negative pressure and it tends to open. And because it's open-ended to the nasal cavity, if there's something there, like for example, some molecules, some urine of another animal that's in the nasal cavity, it will be sucked into the lumen, which contains the sensory neurons, or the sensory endings of the sensory neurons. So this layer right here, I didn't mention that, contains the sensory neurons. So the constriction of the blood vessel, as is shown here, will cause the expansion of the lumen and uptake of stimuli to the glomerulonasal organ and to the sensory neurons of the system. Okay, so this is the process of stimulus delivery, which is analogous or it's related to the process of breathing in the context of the main olfactory system, except that this suction doesn't happen all the time. It's not an ongoing rhythmic process, but it's, as far as we know, it's something that's associated with arousal. So when an animal is in a specific behavioral state, in the case of mice and other rodents, it can be social arousal, or maybe actually some fearful thing in the environment. This will lead to activation of the pumping and to uptake of stimuli to the vomeronasal organ, which is called also VNO, and I will use this term. Okay, and this is actually occurs, as far as we know, under sympathetic control of the sympathetic nervous system. Yeah.
Yeah, that's what I want to ask. Like, if you can control the intake of the stimulus.
The what?
So the intake of the stimulus
is sympathetically controlled. Yeah, yeah. And
So, I was wondering if it's correlated to the heartbeat,
the heartbeat will induce small pulsations here, but I don't think that's a good question. I don't think it will suffice to actually induce suction. You need more extreme changes in the actual diameter, right? It's not the pressure so much, but the diameter of this blood vessel. Yeah.
So it's only, I mean, there's no voluntary way to control it?
For the mice, or for maybe there is, or for the animals. No, I'm not saying. We don't know enough. We don't know about all. I don't know if it's voluntary or not. And there are actually a very small number of experiments because it's very hard to make such recordings. What is known is that there's more pumping activity when actually it's hamsters are in a new environment. I don't know what animals feel, but wait, one thing is very important. You know of the Flamen response that you sometimes see, like horses. It's very famous in horses that they kind of lift up flare their nostrils, and also cats do it. If you look up Flamen, it's Flehmen. Many animals do it, and that's it's an outward expression of suction of fluids, of stimuli to the VNO. So definitely, yeah, animals do have voluntary control of it. Okay, like, and you can see it in cats and horses. I don't know about dogs, but dogs have the organ. I don't know about the Flamen in dogs. Okay, so let's talk a little bit more about the sensory neurons. Not a whole lot, but we have also VNO receptors, and there are actually two major classes of sensory neurons, which we won't discuss their properties. And you can see that actually even at the level of the VNO itself, so this is the VNO, right? You recognize it. This is the blood vessel, this is the lumen, and this is the sensory epithelium. And this is just to show that there are two distinct layers, the basal and the apical layer, and you have different types of sensory neurons in them. And they all project, actually they project to distinct parts of the olfactory bulb, but this is not something that we'll discuss. And as I mentioned before, the sensory neurons, their axons project to the bulb, but not to the main olfactory bulb, but to this smaller region, which is called the accessory olfactory bulb. So that's another important term to remember.
Are these neurons sensitive to different molecules that in the...
Yes, they may sense different molecules. So it's not exact. So it's different types of receptors, different molecules, and there are some overlaps. There are molecules that can be detected by both of the systems, but there are molecules that can be detected by only one and not the other. So there are different receptors, and generally there are different molecules that can be detected by each of the systems. One thing that we can already appreciate is that for molecules to reach the VNO, they actually don't have to be volatile. But they do have to be soluble. So for example, if there's some big molecule in another animal's urine and it's somehow uptaken into the nasal cavity and to the VNO, then it will be perceived by the system. But it won't be perceived by the main olfactory system if it is not volatile and able to reach it. So it's not just the ability of the sensory neurons, but also the physical properties of the stimulus itself that determines which system will be able to detect it. So these are just the structure of the receptors, which I won't discuss, and this is just the fact that they are. We have two different subdivisions. Yeah.
I didn't really understand what you mean by volatile.
Volatile, that they nedifim, that they are they can reach the nose from another. So there may be some stimuli that an animal has, like some big proteins, but they just won't fly. They won't reach me by air. I have to go to the other animal, actually touch it, and get it into my nasal cavity, if I'm a mouse, in order to perceive it.
So if you bring non-volatile stimulus into the nasal cavity, then the main system will not affect.
Much less so, because you're right that some of them, some of it may touch, but there's actually pretty elaborate structures here that cause the fluids to flow down into the floor of the nasal cavity. But you are right that if there's a lot of it enters here, some of it may also reach the olfactory epithelium, but not with the same efficiency as it will reach the VNO.
In the sensory epithelium, we have both the VNO and the olfactory
No, we distinguish between the main olfactory epithelium. Both are in the nasal cavity, but we distinguish it between the main olfactory epithelium, which is shown, which would be here, and the VNO, which is a distinct enclosed structure. So a few words about the physiology of the system. In the system, our understanding of the system lags a lot compared to the main olfactory system, because for many reasons it's harder to study it. This is one example of recording a multi-electrode array. So it's an array of electrodes, and you can lay this can be used for various types of brain preparations, but it can also be used for the VNO. So you can take out the epithelial layer within the VNO and then measure the responses. And this is just one of the first studies showing a systematic presentation of, in this case, urine stimuli, of female urine and male urine, and showing that there are neurons that respond in a concentration-dependent manner to the male and the urine stimuli. So this was just a demonstration, initial demonstration of the physiology of the system, and showing also that it corresponds to our understanding of the system as related to social interactions, because urine, as you probably know, is a very important source of information from one animal to another. There's a lot of information about the sex and the species and the strain of the mouse and so on. Yeah.
But here the response was not specimen specific, so they tried urine of different females, because we talked about pheromones, and here I would think it's within species and not specific, but could the VNO be used to specific identification?
So I think in these studies there are actually mixtures of female urine, but there are I'll show you some studies actually that also look at individuals, and there is an ability also to distinguish between different individuals by the system. But when what?
So there could be some learning.
There could be. Yes, definitely there is what to learn there. It's not just stereotypic molecules, that are detected there. I want to mention two. So one of the questions was, what are the ligands of the system? It's something that also you brought up, and there are two classes of ligands that I want to highlight a little bit, because they're interesting. One of them are called the major urinary proteins, or MUPs for short, and these are big proteins that are secreted in the urine of mice. In humans, we don't secrete proteins normally, but other animals do. And what these images show you, these are biochemical separations on a gel, and each band shows you one type of protein. The point here is that different mice have different combinations of these major urinary proteins, of these MUPs. Okay, each one is one MUP. So an animal, there's about 20 different MUPs overall, and different strains of mice or different individual mice in the wild, each of them will have a different combination of MUPs. So maybe if I'm a mouse, I will have MUP 1, 7, and 12, and my friend mouse will have 8, 9, and 16, and this can people think of this as kind of like a barcode that can provide the identity of different individuals. So this is something, and the nice thing about this is because they're proteins, they're genetically encoded in a simple way. So you can think of some way that these MUPs may encode individuality of mice. Yeah.
Is it consistent over time?
Yeah. It's consistent over time in diet. It may change with the hormonal status, so it may, in general, it may, all of them will increase or decrease it, but it's not that one, as far as we know, there's not a modulation of specific, no change in combination. Okay, this is just an image from a study that shows that the MUPs, some of the MUPs can mediate aggression. So we talk about pheromones and reproduction. Here's an example, if we want, we can call this a pheromone, that if you take this isolated protein and you put it on a castrated male, which usually doesn't elicit aggression, it will elicit aggression in another animal. So this, we know that some of the MUPs have the feature that if perceived by another mouse, they will evoke aggression. Maybe telling it, the mouse, that now a male came into my territory and...
This will evoke the aggression in the castrated?
If you take a castrated male, which usually doesn't, so not aggression by the castrated, but towards the castrated male. Another class, which I'm mentioning also because it's the subject of one of the papers, are the sulfated steroids. So steroids is a rich class of molecules that they serve roles in coordinating physiological processes in the body, and to be secreted in urine, they're often chemically modified so they can be soluble, and often in mice they're sulfated. So by sulfating them, they become soluble in water and urine, and then they're excreted. And it has been discovered recently that there are many responses or many sensory neurons in the vomeronasal system that respond to these sulfated steroids in a molecule-specific manner. So these are examples of different steroids with the different classifications: estrogens, progesterones, androgens, and here are different cells. And what this matrix shows that different cells have differential responses to each of these individual stimuli. So I described two classes of chemicals, the MUPs and the sulfated steroids, and there are few other examples of known ligands of the system, but it's not like we know all of the ligands, we know some of those. Okay, that's what I just said. The search is gone, still goes on. One word, one slide about signal transduction. If you remember, what was the molecule that elicited opening of channels in the case of the male factory system? Do you remember? The cyclic AMP, right? We had this cascade. The receptors activated the G proteins, which activated the adenylyl cyclase, which increased the concentration of cyclic AMP. Here, there's a different transduction process, and also the receptors are G protein coupled receptors. They activate G proteins. It's a different signal transduction cascade based on phospholipase C. That's the enzyme that cleaves the steroids in the membrane. But the point that I want to make, which is relevant for the next slide, is that there's a different ionic channel that mediates the depolarization that activates these neurons. This is a channel called the TRPC2 channel. You may have heard of TRP channels. So the TRPC2 channel is maybe not exclusively, but it's mostly expressed in vomeronasal sensory neurons, both types of vomeronasal sensory neurons, and it's not expressed or almost not expressed in male factory system sensory neurons. So this provides a genetic way to the expression of this channel provides a genetic way to modulate, and as I will show you just now, to perturb, to destroy the activity of sensory neurons in the vomeronasal system specifically. And this is what was done by now. It has been done in several studies, and various outcomes have been tested. This is one. These two studies provide the first examples of this manipulation. So these are transgenic mice in which the TRPC2 gene has been knocked out, so they're not supposed to have the ability to relay information from the VNO, right? The sensory neurons are not functioning. And so here you see, let's first look at the left column. So this is what each of these ticks correspond, or the behavioral assay is you have a mouse, a male mouse in a cage, and another male is introduced. Okay, and the response of the resident towards the intruder is quantified, and it's quantified by these little ticks. Each tick corresponds to one bout of aggression, one attack by the resident towards the intruder, and each row is a different trial, so it's been repeated, this experiment, many times. Now, what you see here is the response, the aggressive response of a male mouse towards a castrated intruder. And you can see that there's not a lot of aggressive responses, and this is most meaningful when you compare it to what happens when you give a castrated mouse with male urine on it. So the male urine has been swabbed on the poor castrated mouse, and then when he's introduced to the cage, the resident attacks him much more actively. You can see it here, right? Much more black ticks. So this is known. This is nothing this is the baseline. The interesting manipulation is this one. It's a very boring graph, but what it shows you that that the residents, if they don't have a VNO, then what? They don't attack. They're not aggressive towards other males. So this is one thing that's very prominent about these mice. And these are two studies which actually came out at the same time almost and showed the very similar things. I'm showing one aspect from each study. This looks at sexual behavior. And here it's again the baseline. It's male-female mounting. Okay, so you quantify how much the male mouse mounts and intromits with a female, and when you compare the control mice in red and the TRPC2 knockouts in blue, there is no significant difference. So a male, you don't need the VNO actually to mate with a female if you're a mouse. The interesting difference comes here when you look at male-male mounting, which is something that mice rarely do. Male mice rarely mount other males. This is shown by the control bars, which are low. However, the TRPC2 knockouts, without the VNO, they mount the males as frequently as they would mount the females. Okay, so there's it's not exactly the same, but they mount. There's a lot of male-to-male mounting. So without a VNO, the male mice don't have. Presumably, there's some inhibition which normally prevents them from doing it, and now they do it. And this has been, as I told you, this has been shown in both of these studies, and there have been other studies, and it was shown that when you knock out the VNO in females, it makes them, if you've been to Tali Himchi's lecture last week, then you know about, I think she mentioned also this specific aspect. It's her work showing what happens when you knock out the VNO in females. They start behaving in male-like patterns. But it's also important in parental behavior, in predator avoidance. So mice without a active TRPC channel will be much less anxious and will avoid predator odors much less than normal mice. So it has a general role in many such social or behaviors that we think of as innate behaviors important for survival. And again, this is not to say that the male olfactory system doesn't, but it just tells us that it probably is a key role, or it's definitely a key role of the glomerulonasal system. So let's now move on to the next stage. After talking about the sensory neurons of the system, I want to talk about the connectivity in the accessory olfactory bulb, which I remind you is a small region at the back of the main olfactory bulb, which receives information from these sensory neurons. And here we saw this organization, right, that all the sensory neurons converge to glomeruli, and you have a mitral cell that each mitral cell samples information from one, or samples the axons of one type of sensory neuron. In the accessory olfactory system, it's different. Although a given glomerulus does receive information only from one type of receptor, this is still true. The difference is that a given mitral cell can sample information from multiple glomeruli, and there's a lot of heterogeneity. So some mitral cells may sample only from one glomerulus. And some other, for example, the ones shown in E and G, look how many different how many different glomeruli they send their dendrites to. And it seems like that we have here a connectivity that allows mitral cells at this stage already to obtain more complex representations of combinations of sensory neurons, because each glomerulus is still one type of sensory neurons. Now, what is the functional implication of this? It's something that we actually do not fully know. It could mean that the mitral cells will only fire when all of the glomerulus are active, right? It could be some kind of an AND operation, but it also could be some kind of an OR operation, meaning that the mitral cell may be active when each of these, when glomerulus is active. So we don't really know, but we know that it's different, and it can give rise to something that's a bit more complicated at the level of the olfactory bulb. This is the first paper that actually recorded the signals from the accessory olfactory bulb, the electrophysiological signals from the accessory olfactory bulb in behaving mice. This was done in the lab that Adi Mizrahi and I did our postdocs. This is actually the paper that caused me to do my postdoc in that lab. Shows two mice interacting, and what you see in this recording is a trace, a voltage trace recorded in one mouse. You see the mouse here. This is a mouse that's investigating another mouse. This mouse is under anesthesia, so it's not moving around. And you can see that this mouse is investigating the arena, and there are certain epochs where it's investigating the rear, the anogenital region, or the facial region of the anesthetized mouse. And you can see that there is an increase in firing rate in this specific cell when it investigated mainly the head region, but also the tail region of this mouse. So the point from this study is that it was the first study actually recording activity in behaving animals, showing that indeed cells in this region care about other animals. So again, supporting the role of this region in social interactions. And it also showed that there's selectivity. So you cannot see it from here, but if you record from one cell, it may respond only to females, or only to males, or only to males of one strain versus males of another strain. So there's the ability of cells at this stage to distinguish between different types of animals. One of the drawbacks of this kind of approach is that you don't really know when the VNO was activated, because we don't have a direct measure to know when there's actually uptake, when there's actually suction, and we also don't know what exact ligands activated the neurons. Right? In the facial region, there could be various sources of stimuli, saliva, or maybe tears, or something else. And the same for the rear region. So this was a very important study, but it had the limitation of not allowing a systematic analysis of the system. And this is the type of experiment that we're doing in the lab, which is again recordings from the accessory olfactory bulb, but we're doing them in anesthetized mice. And the challenge, of course, is to activate the VNO, right? Because I told you that the VNO is not active all the time. It's active under states of arousal. But I also mentioned that it's activated by the sympathetic nervous system. And the approach that we use in the lab is to artificially stimulate the sympathetic nerve trunk, and that we found out that a long time ago, but it actually activates the vomeronasal organ and can induce suction. So when we do an experiment, I will just describe this quickly. Slide, there are various stages, but the main point here is that when you give a stimulus to the nasal cavity, initially, and a stimulus could be, for example, a drop of urine. Initially, there's no obvious change in firing rate of a given cell that you're recording from, but once you activate the sympathetic nerve trunk and induce suction, this is shown here by the expansion of the lumen, you will see an increase in firing rate. If you're lucky to record from a neuron that responds to this specific stimulus, because not every neuron will respond to every stimulus. Okay, is this clear? You're with me? Good. So, yeah.
So it just stimulates the
happens due to the sympathetic activation. Yeah. First we put some urine on the nose, then we activate the sympathetic nerve trunk, and that sucks the stimulus in if everything is working correctly. And this is an example of such a recording where you have many trial repetitions, not that many, but 6 different trial repetitions and recordings from 2 different neurons, showing you. I will start accelerating a little bit now. Showing you that these are PSTHs, and you have a green neuron and a yellow neuron, and what you see here is that there is selectivity. The green neuron responds to female urine but not to male urine, and the male urine responds only and the yellow neuron responds only to male urine. So you have sex selective responses. There's not much new about this based on what you saw before, except that now we know we have a specific stimulus that we isolated. And okay, before I go on to show you some more refined examples, I want to describe the process of the Bruce effect. And this is also a depth that I have for describing pheromonal process, a pheromonal response that is a primary response. Remember when I classified, I said there is a releaser response, and there's another one, a physiological response, and here's an example. And the Bruce effect is a phenomenon in which a female mouse, a pregnant female mouse, when she's exposed to a strange male, so the father in this case is shown in brown, so there's a brown litter here, and then she meets a strange male shown here in black, and with very high probability this will terminate the pregnancy. This is called the Bruce effect or the pregnancy block effect. You may have heard of this before. One of the really nice things about this effect is that the father itself will not cause pregnancy failure. So during mating, the female forms some kind of a memory of the mating male, and this prevents that male from eliciting the pregnancy block. And this is one of the things that we're actually interested in. This is one example of plasticity in the system. But the reason I'm showing this, first because it's cool, but also to show that in order to be able to mediate this effect, the female mouse has to have the ability, or the neurons in the AOB need to have the ability to distinguish between different individuals, right? Otherwise, how can it distinguish between the brown and the black male? I'm not going to talk about the cascade of events that elicits the pregnancy block. There's just not enough time for that.
Does it have a function? I mean, that there's a pregnancy failure, or is just
okay, this is a good question that's often raised. There is a benefit to the. You can think of a benefit to the male and to the female for the pregnancy block. So to take it to extreme, in the case of the female, maybe the incentive is not if there's a more dominant male came, okay, and throughout the previous father, it may be better to invest her resources in the litter of the new male rather than the old male, rather than the weaker male that she escaped. We can talk about this after the class. There's a long answer to this question. For now, I have only a few minutes left, and I'd like to show you that there are responses. When you look now at 3 different neurons, we find neurons that are selective for a specific combination. For example, here this is one neuron that responds to white. White is just an indication of the strain of the female. So this is a very selective neuron, and here's a neuron that responds to to all the males, regardless of their strain. So we have various types of responses in the accessory olfactory bulb, some of which allow us to distinguish between different individuals. Okay, the one other thing I wanted to show you here is that the neurons in the vomeronasal system, in the AOB, they don't just respond to stimuli from other mice. So it's not just about social interactions, but they also show very strong responses to predator urines, indicating that they may have a role in predator avoidance. And we can also see the different neurons, different individual neurons can distinguish between different individual predators. So one neuron may respond to a fox urine but not to a rat neuron, whereas some neurons may respond to rat but not to bobcat or to fox. urine. So this system is not just confined to stimuli from other animals within the same species. It can also act across species.
Neural response for enemies also shows up in behavioral?
Yes. You mean, do they avoid them? Definitely yes.
You just give them because I suppose these rats or mice were never exposed to the actual predator. So, but they still take if you expose them to the urine, they will feel like.
Yeah, it's an innate avoidance that they have for these different stimuli. Yeah. I can do this really quickly just to give you the main points, and I'm not going to expect you to remember this, but I want to go over this really quickly. So there are some neurons in the AOB that respond both to predator and to female urine, for example, and that's very problematic, right? Because if you have one neuron that responds to both of these stimuli, and you look just at this one neuron, you won't be able to know whether you need to mate or to escape from that stimulus. And the question is, how do downstream regions distinguish between the two types of stimuli or the different types of stimuli? And what I won't have time to show you is that if we look at more advanced processing stages, if we look at the medial amygdala, which is the region that receives information from the accessory olfactory bulb, we'll see that there the responses are more selective. So a given neuron will respond to a predator or to a male more selectively than at the previous stage of the AOB, where responses may be more homogeneous. There's still a distinction, but it's sharper at the level of the amygdala. The other thing that I would have shown you about the amygdala is that we have actually a sexually dimorphic pattern of responses in the medial amygdala. So male and female amygdalas, at least in mice, are different. The female amygdala are much more, or the responses of neurons in the amygdala of females are much more sensitive to urine of other males. Okay, and in male amygdalas, the neurons are much more sensitive to the responses of other females. So there's a sexual dimorphism that develops at the more advanced processing stage of the amygdala. It does not appear at the level of the AOB, at least it's not prominent. Okay, but this is one example which is also connected to the talk that was given here last week or 2 weeks ago about sexual dimorphism and the electrophysiological correlates of this. This is a very light slide. This always comes up, the issue of human pheromones and the question whether humans have pheromones. And the question is how we define pheromones or not. This is one example. There are many examples of people or companies who market pheromones that are designed for males and females, and there are also pheromones for males wanting to attract males, females wanting to attract females. I mean, all the 4 combinations you can buy, not necessarily from this company. And I don't think that there's a lot of good evidence to support these products, so I wouldn't spend my money on this, although each of these companies is usually has some reference which shows some study that was done some time ago and shows a difference based on the pheromone. So on a specific molecule, and it can be molecules that worked in mice or in other animals, and they're usually also marketed as human pheromones. So I'm saying I personally don't think that there are any molecules that once you perceive them, you lose control, and I don't even think that they change your sexual behavior very much. But I cannot be confident about this. This is my feeling from what I know about the system.
It's related to the fact that we are more. We care about being clean much more than animals. They shower.
Is it related? There's a whole discussion about the human maybe fear of our own real smells, and so yeah, I don't know if it's related to this or not, but in humans definitely we're much more. We're less affected, or our odor environment is much more artificial than what it was originally. Yeah.
there are societies where human males consider themselves.
You're asking me if there's such societies? Yes, I think also, you mean body odors, very strong body odors? I think so. I think that in some places, I think of Asia actually, that people are, or at least it's not much of a taboo to have normal body odors. Yeah. Okay.
What about some of like non-SOBIS research that has to do with human pheromones or something?
Yeah, so there is some evidence of that, but the effects are usually pretty minor if you look at them closely. So both the physiological effects, mostly the physiological effects, also the behavioral, there are effects. So that's why I'm not giving a concrete. I don't want to be too assertive about this. I'm just saying that I don't think there's anything that's very, very strong that acts in a very clear way, as we may see in insects and some even in some rodents. Some examples that I did not even give. I won't go over this. You can look at it later. It shows you the distribution of sensory of vomeronasal receptors in different species. It's interesting that humans do have some V1R receptors, even though we don't have a V and O, and presumably they're expressed in the manofactory epithelium. And I just wanted to show you, this is the last slide that I want to show you from a very recent study that just came out, so I thought it would be nice to mention it. And people looked at the sequence of this receptor, and they looked at specific polymorphism. So an amino acid change that has a function, that supposedly has a functional implication on the function of the receptors. We don't know, of course, what it really does to the receptor, but there is a difference, a genetic difference. And what they did in the studies was to test the correlation between this genotype and sexual behavior of people, as reported by a long questionnaire. And there is some measure that they call here the social, I forgot the social something index. No, social, but the higher the score, the questions were how many sexual partners did you have, will you have, do you think, or how many one-night stands. These were the questions that brought up to this score. And in females, but not in males, there was a certain correlation between one genotype and the answer. So one genotype was correlated with more socio-sexual behavior, okay, as expressed by these answers. You can see that there's a lot of variability here, right? You see the error bars are very, very, very big. So again, and it's hard for me to think how exactly this would work, but there are indications that there are some effects, and it could very well be that other sensory receptors might have similar effects as well. But so this is kind of the state of the art about what we know about the role of sexual behavior and olfactory reception in humans in this way. With all that said, I think it's very clear that odors of other people can affect how much we want to be close or not close to them, right? Regardless of whether we have pheromones in the same way that I described before. So that's it. Sorry for taking you over time.